Welcome back to Bioinfo. In today's explainer, we are doing something really exciting. We're going to transform raw, invisible biological data into absolute cinematic visual insight, and we're using PyMol to do it. It's the ultimate tool for this. We're going to follow the complete journey of a protein, taking it from a basic string of letters to a dynamic 3D reality.
Okay, let's dive into this. If you think about it, before PyMol, structural biology could feel pretty intimidating, right? You're staring at these endless sequence codes and really confusing, tangled gray wireframes. But after PyMol, it's a completely different world. You get these vivid, publication-ready, ray-traced insights. We're talking deep navy and teal environments that just beautifully illuminate those protein domains. It literally makes the invisible completely readable.
And the best part is, getting started happens in milliseconds. It's just a rapid-fire sequence of operational steps. You type fetch 1HHO to instantly pull your atomic coordinates straight from the protein databank. Then, orient squares the molecule up perfectly on your screen. Finally, hide solvent strips away all that distracting water. Boom! 3 quick commands and our canvas is clean and ready to go.
Let's move on and see how this builds. Now we get to choose our lens, because PyMol gives us 4 core display modes. You've got cartoon for secondary structure, surface for interaction contours, stick for chemical side chains, and sphere for the true physical volume. And the key here is that each of these modes reveals an entirely different structural story. For instance, in cartoon mode, we strip away all the atomic noise to highlight the protein's structural core. We're talking about the pillars of life here, the alpha helices and beta sheets. You know, these aren't just random floppy loops, they are highly precise 3D configurations securely locked together by hydrogen bonds between neighboring amino acids. But if you switch to surface mode, our perspective completely flips to the exterior. This is literally exactly what an incoming drug or a water molecule sees as it approaches. It highlights all those deep crevices, the exterior pockets, and the exposed side chains that are absolutely vital for chemical binding. So the crucial point is that to truly read a structure, we have to map physical data using color. We're not just making it look pretty, we're giving it meaning. By sweeping vivid colors across the molecule, we can distinguish secondary structures, separate hemoglobin's 4 distinct chains, visualize temperature and mobility via B factors, or even map out hydrophobicity to spot those oily membrane regions.
Now, what's really interesting about this slide is a reminder that great science communication absolutely has to be inclusive. Standard red-green coloring can be completely invisible to colorblind researchers. That's why PyMol includes the J-Fly colorblind-friendly Okabe and Ito RGB palette built right in. It uses these highly specific values for sky blue and vermilion, ensuring your visual insights are crystal clear to everyone.
All right, let's ground ourselves in the biochemistry of the active site. When you zoom in, the whole game comes down to hydrogen bonds. These are transient, non-covalent bonds. Super tiny, just about 2.7 angstroms in length. But these precise little connections are the ultimate key to ligand interactions, dictating whether a life-saving drug locks in tight or just floats right on past. So how do we analyze them? We isolate these micro-interactions. Using the command select binding site, sidechain within 5 of res J9U, we can instantly ignore thousands of irrelevant atoms. It basically draws this perfect 5 angstrom spotlight right around our target ligand and its immediate environment.
With our target zone isolated, it's time to measure. The distance command lets us calculate the exact space between very specific atoms, like between a nitrogen and an oxygen. PyMol then visually displays those critical hydrogen bonds as dashed lines, giving you absolute visual proof of that interaction.
Okay, now we're ready to render. To get that publication-ready figure, you walk through a very specific sequence. You perfect your camera angle, drop the background color to white, turn the opaque background off for total transparency, and then fire off Ray 2400-2400. That triggers PyMol's massive high-resolution rendering engine.
And this brilliantly illustrates just how powerful that ray tracing engine really is. You've got 4 distinct aesthetics to choose from. Mode 0 gives you stunning photorealism. Mode 1 adds crisp black outlines. Mode 2 strips it down to a stark technical wireframe. And Mode 3 delivers this really cool quantized, stylized look. It's perfect for matching whatever style your target journal requires.
But here's a little pro secret. Professionals almost never rely on manual clicks. It's all about scripting. Writing scripts guarantees 100% reproducible figures for every complex shot. It automates all your repetitive rendering tasks, integrates seamlessly with Python, and lets you email your entire 3D workspace to collaborators as a tiny, lightweight text file.
Seriously, watch how incredibly fast this is in practice. A single line of code can fetch a brand new structure, instantly apply fancy tubular helix aesthetics, and uniquely color a ligand in milliseconds. It turns dozens of tedious manual clicks into one flawless keystroke.
Now, let's push beyond static images. PyMol actually doubles as a cinematic animation studio. We can animate continuous motion across 4 distinct dimensions. You can sweep the camera view, move object positions to show ligand dissociation, morph molecular states, and even execute commands directly on a timeline. Building this motion is all based on keyframes. You use mset to build out the length of your timeline. Then, mview stores snapshots of your camera and objects, while PyMol automatically and smoothly interpolates the kinetic motion between them. mdo handles timeline commands, and finally, mpng exports every single frame for your video.
So what's the ultimate cinematic shot? For sure, it's a multi-stage structural morph. We put it all together by aligning an open and closed crystal structure, and then executing morphs to molecule. PyMol calculates all the intermediate kinetic steps, basically simulating the protein physically breathing and shifting conformations right in front of you. Load, color, analyze, publish. From raw, intimidating data to dynamic cinematic molecular movies, PyMol puts ultimate structural control right in your hands. You've got the commands, and you know the workflow. So what hidden structural masterpiece are you going to visualize next? Thanks so much for joining us for this explainer.